Hello everyone, welcome to our new video. In today's video, I am going to discuss some basic questions about respiration. Respiration is an catabolic process. It means it is degradation of organic compound to give energy. You can also try to answer these questions. I hope you will learn definitely something from this video. Let us start with our first question. Energy currency of cell is ATP, PPP, protein or fat. This is very basic question. We all know that ATP is the energy currency of cell. So right answer is A. Let's move to next question. The primary and instant source of energy is organic acid, fat, protein or glucose. When we eat food, which substrate gives energy primarily? And it is glucose. So right answer is D. If the question is which substrate gives highest energy, then answer will become fat. Fat is the highest energy giving biomolecule. Next question. In long period of fasting, what will be the sequence of substrate used up by the body? And this is the different combinations of substrate that can be possible. We have to see here the correct sequence of substrate that are used by body in long fasting. First of all, it will be carbohydrate, then fat, then protein. So correct answer is C. Next question. Respiration which involve protein as a substrate is known as. When respiration involve carbohydrate and fat as a substrate, then respiration is known as floating respiration, and when it involves protein as a substrate, then it is known as protoplasmic respiration. So right answer will be B. Next question. Gaseous exchange occurs in plant through stomata, lenticels, C both or D none. Stomata are the pore-like structures that are present in green parts of plant, and lenticels are also raised pore-like structures that are present in woody parts of plant, woody parts. So these both perform gaseous exchange function. So correct answer will be C, both. Next question. Which of the following enzyme is localized to inner mitochondrial membrane? Options are malate dehydrogenase, succinate dehydrogenase, fumarase, D both A and B. Succinate dehydrogenase is the enzyme that is attached to inner membrane of mitochondria. So B is the correct answer. Next question. In absence of oxygen, consider the following point about cell. A. Citric acid cycle and oxidative phosphorylation cannot operate. B. Supply of NAD positive become limiting. C. Glycolysis may stop. D. Cell undergoes fermentative metabolism. And we have to choose the correct combination. So all these events can happen when there is no oxygen in the cell. So right answer will be fourth, all of the above. All these processes will operate here because NAD positive supply is limiting, and once all NAD positive get converted into NADH, that is its reduced form. Now, the reaction catalyzed by enzyme glyceraldehyde 3-phosphate dehydrogenase cannot take place. To solve this problem, plant will undergo fermentative metabolism to regenerate this NAD positive and will help the cell in surviving anaerobic conditions. Next question. Pasteur effect is production of ATP in Krebs cycle, B. Increased rate of glycolysis in anaerobic respiration, more energy production in aerobic respiration, or none of the above. Pasteur observed this effect in yeast cell. When yeast cell transferred from aerobic to anaerobic condition, there was increase in rate of glycolysis. So correct option will be B. Why there is increase in the rate of glycolysis? Because in anaerobic respiration there is lower energy production, and for survival of cell higher energy is needed. So rate of glycolysis will get increased. Next question. Glycolysis is also known as EMP pathway, CTC pathway, hexose monophosphate shunt pathway, or D, both A and C. Correct answer will be EMP pathway. Why? Because of its discoverer. Their name is Embeden, Meyerhof and Parnas. Next question. During seed germination, gluconeogenesis occurs in A, ribosome, glyoxysome, cytoplasm of young leaves, or D, mitochondria. What is glyconeogenesis? It is the formation of glucose from the substrate other than carbohydrate, such as organic acid or fat molecule or organic acid. In germinating seed, it occurs in glycosome. So right answer is glyoxysome. Now let's have a look on some spicy points. Phosphatidylcholine or phosphatidylethanolamine are important lipids that are present in mitochondrial membrane. Second point, cristae are the invaginations present in inner mitochondrial membrane for increasing the surface area of membrane. Next point, control of glycolysis is bottom-up control. Why it is bottom-up control? Because primary regulation is at the level of phosphoenol pyruvate by pyruvate kinase and PEP carboxylase, and secondary regulation at the role of enzyme phosphofructokinase. So their sequence is from bottom to up level. That is why it is bottom-up control. Next point, PDH complex reaction act as connecting link between glycolysis and Krebs cycle. Glycolysis product, that is pyruvate, act as a substrate for the PDH reaction, and its product act as substrate for Krebs cycle. So this is known as connecting link between both. Next point is EMP pathway. Glycolysis reactions were given by Gustav Embeden, Otto Meyerhof and J. Parnas. So this is EMP pathway. That's all for today's video. Now I want to ask a simple question from this video. Name the enzyme that is present in mitochondrial membrane. You can answer this question in our comment section. Hope you enjoyed the video. You can also tell me on which topic do you guys want next MCQ video. Thank you so much. Bye bye. Take care.